Thank you very much for your kind introduction, Bijan. A big thank you also goes out to biocrates and ASCA Medical for the opportunity to share my research with you all. So today, I will talk about the fascinating world of metabolites, but specifically metabolites of microbial origin. And I want to start highlighting what my group does. We investigate metabolites produced by food-gut microbiome interactions, and we also study the impact of these metabolites on the intestine. And this is what I will present today. Today, the first half of my talk, I will present recent research work where we investigated food-gut microbiome interactions, paying particular attention to microbial metabolites, and secondly, the models, cell models that we use to assess to determine the impact of these kind of microbial metabolites in the intestine. When we talk about food-gut microbiome interactions, there are many aspects that we should consider, but among them, dietary patterns, the way that we eat, and also the amount of food that we eat must be considered. So if you keep in mind that we eat approximately 1 to 2 kilograms per day, that's over 400 kilograms of food yearly, and approximately 30 tons over the course of a lifetime. That's a huge amount of food that makes its way through the digestive system. And the digestive system, the main function of this system is to transform the food that we eat into nutrients and energy to keep us alive. And the gastrointestinal tract, apart from transporting and transforming food that we eat, is colonized by microorganisms that live in symbiosis with the host. These gut microbes utilize digested and undigested food as an energy source to promote gastrointestinal health, but also to keep this symbiotic relationship. And among these metabolites, these regulations are coordinated by many metabolites that can interact with intestinal epithelial cells and activate or inhibit specific molecular pathways that in turn promote gut health or trigger gastrointestinal diseases. So delving deeper into this concept, my research aimed to transform our understanding of the food-gut microbiome intestine triangle, and paying particular attention to the pivotal role of microbial metabolites. And this involves first deciphering the impact of dietary factors such as macronutrients, phytochemicals, food processing, and particle size into production of gut microbial metabolites. Secondly, our goal is to investigate the biological role of these microbial metabolites on the intestine. And lastly, we plan to use this knowledge to develop novel foods and dietary strategies to promote gut health and prevent gastrointestinal diseases. But let's begin now talking about gut microbial metabolites, and particularly short-chain fatty acids. So during the digestion, simple sugars are rapidly absorbed in the small intestine. However, complex dietary fibers reach the colon nearly intact. And in this region, dietary fibers are utilized by microorganisms as a source of energy, releasing in the lumen short-chain fatty acids. So these short-chain fatty acids can stimulate the release of gut peptides, such as peptide YY, GLP-1, GLP-2, interacting with the G protein coupled receptor on endocrine cell lines. Other short-chain fatty acids, such as butyric acid, promote barrier functionality and also support the control of anaerobic conditions in the colonic region by activating beta oxidation in mitochondria enterocytes. And in the vast majority of cases, short-chain fatty acids are associated with the intake of dietary fibers. The higher intake of dietary fibers, the higher the amount of short-chain fatty acids that are being released. However, have you ever wondered if your eating behavior modulates gut health? With this research question, we wanted to determine if the particle size of those particle size serve as a critical abiotic factor in modulating the production of gut microbial metabolites. And the field of oral processing behavior has already reported that fast eaters generate large particles, and by contrast, slow eaters generate small particles and therefore a larger surface area. And several nutritional studies have also reported that the size of these particles affect the absorption of nutrients and also the release, for example, of insulin. However, it remains unknown how these particles, the size of these particles, modulate the upper and lower intestines. And we hypothesize that the size of these particles can modulate microbial communities and in turn the pattern of microbial metabolites that are being produced. And we target initially short-chain fatty acids. So our Ph.D. student, Jen, designed this experiment. First, we selected 3 kinds of rice with different fiber content. Then firstly, we cook it. And after that, one participant masticated the rice 5, 10, 20, and 40 times. And then we characterize the properties of this bolide in terms of particle size, surface area, and saliva intake. The bolide were then digested, and at the end of the digestion, again in the digester, we characterize the particle size, surface area, and start hydrolysis. And the undigested fraction was used to perform fecal batch cultures at the end of this experimental design, and we monitor again particle size and short-chain fatty acids. So after mastication, as expected, 5 chews generated a small number of particles, and those particles were larger compared to 40 chews. And 40 chews generated a significantly higher number of particles, but those particles were smaller. And the total surface area follow a linear behavior with the number of chews. As soon as we characterize the bolide, then this bolide were digested following the InfoJust protocol. And at the end in the digestia, we look again for the number of particles, particle size, and total surface area. And we notice that this process significantly affects the number of particles and the size of these particles. And we decide to use 5 chews and 40 chews in our bioreactors. Because as you can see in this graph, there were statistical differences between 5 chews that somehow mirror fast eater and 40 chews that recapitulate a slow eater. And of course, a picture is worth 1,000 words. You can see now on the left side, the basmati brown rice that we use that was masticated 5 times and digested. And in this case, you can clearly see that the particles are larger compared to 40 chews. And if we look now on the left side, where the basmati brown rice was masticated 40 times and then digested, those particles were smaller. And those are the particles that in this experimental design were exposed to the gut microbiome. So having established that, differences between 5 and 40 chews at the end of the digestion, we ran fecal batch cultures. And first, we look at the number of particles that remain unchanged over time. However, we notice that the total amount of short-chain fatty acids differ between 5 and 42. 5 choose, therefore, larger particles increase the total concentration of short-chain fatty acids. And now, if we zoom in on individual short-chain fatty acids, we observe that 5 choose, again, large particles increase the concentration of acetic acid. 40 choose increase the concentration, slightly increase the concentration of propionic acid, particularly at late stages of the fermentation. And that's small particles that promote the higher concentration of propionic acid. And butyric acid in this study was correlated with the fiber content rather than the number of tubes or the particle size that we use in the bioreactor. Well, we plan in the future to validate this in vitro study using samples from the Rubert study. In the Rubert study, 40 participants were fully characterized. The oral processing behavior of 40 participants was fully characterized. And in this case, we can trace particles from food to poo. And that's basically what we plan to do, have full control of the particles from food to poo, and then correlate those particles with signatures of microbial metabolites. This dietary intervention would end in November, and we plan to start analyzing those samples at the beginning of 2025. Let's expanding the scope of this presentation. Let's talk about lipids. Well, we know that approximately 5 grams of fat will reach the colon daily in normal conditions, of course. And based on the scientific literature, we also know that dietary lipids are being used by the gut microbiome to produce bioactive lipids that can interact with intestinal epithelial cells and in turn improve barrier functionality. And one of our former Ph.D. students, Josep, investigated in his doctoral thesis the influence of dietary lipids on the microbiome. And in one of his latest works, Josep performed targeted and untargeted lipidomics to look at linoleic acid, conjugated linoleic acid. So Josep first performed GC-fit targeted approach to quantify conjugated linoleic acids. And first, we exposed the microbiome to walnuts, soybean, and sunflower seeds. And we noticed that walnuts significantly increased the concentration of conjugated linoleic acids. You can see here in the red triangle and red lines. And these compounds, based on the scientific literature, for example, in acute colitis in animal models, attenuate the symptoms, and also using 2B immortalized cancer cell lines, those compounds have inhibit the proliferation. Well, subsequently, we performed untargeted lipidomics because we wanted to determine whether other carbon-18 fatty acids could derive from linoleic acid. And we ended up with a list of candidates. And we found that fatty acids with one or several hydroxyfunctional groups that derive from linoleic acid. 2 of those were unambiguously identified, and others were tentatively identified. And I must admit today that it was really challenging because for certain lipids, we couldn't confirm or deny that they derive from linoleic acid. And building upon this challenge, we designed an experiment using isotope labeling. So we use carbon-13 linoleic acid to reconstruct the microbial pathway the microbial biotransformation of this lipid. So firstly, we collected the feces from different donors, and then we exposed these microbial communities to carbon-13 linoleic acid and carbon-12 linoleic acid. We collected samples at different time points, and we performed then isotope tracing lipidomics and shotgun metagenomics. Regarding the lipidomics workflow, we combined MS-style with different databases, and we ended up with a data matrix with hundreds of lipids that derive from linoleic acid. For the entire the microbiome data, this data was processed by the log facility at the Wageningen University, and they provide us to list. One list was related to taxonomy, the other one to functionality, and we are these days at the stage of merging this data and integrating all the data together. So unfortunately, I cannot present preliminary data today, but I must admit that this data is very promising, and we do hope to reconstruct the microbial metabolism of linoleic acid, soon describing the linoleic acid uptake, chain modifications, and synthesis of complex lipids that derive from linoleic acid. And by the way, linoleic acid is one of the most abundant lipids that we can find in plants. Let's transition now to proteins. Well, undigested proteins reach the colon, and then microbial communities can use undigested proteins to produce microbial metabolites. And many of those microbial metabolites derive from amino acids, and they are potent ligands with the aryl hydrocarbon receptor, exerting a potent anti-inflammatory activity and cross-linking the microbiome with intestinal epithelial cells and the inner compartment. In this line of thought, we have recently published a review where we explain plant protein and gut microbiome interactions, paying particular attention to the current state of the art, microbial metabolites that are being released from protein fermentations. And last but not least, we critically discuss experimental diets using plant proteins in order to attenuate gastrointestinal diseases such as colorectal cancer or IBD-IBS. And a couple of years ago, David visited us in the Netherlands, and we jointly investigate the hypothesis that plant proteins and animal proteins generate a different pattern of microbial metabolites. We first selected three matrices. Well, first it was we selected a beef patty, and secondly, plant-based commercial patty rich in pea proteins, and after that we also create our own plant-based patty in the laboratory. Those food matrices were cooked and then digested following the InfoGest protocol. We first characterized the supernatant, what it's supposed to be absorbed in the small intestine, and compared this data with the literature. And secondly, the undigested fraction was used to perform fecal batch cultures, and we took samples at different time points. Data was processed by MS-DIAL, GMPS, and metaboanalysts. And as expected, many gut microbial metabolites derive from amino acids. For example, we noticed that the beef patty will produce higher concentration of indoles. It's a total indole content. What we quantify here, it was a HIT and assay compared to plant-based patties. You can see also on the left side, the substrate, tryptophan, was utilized at the very beginning after 8 hours, 12 hours fermenting. And then the production of indoles significantly increased. So as I already mentioned, it's the total amount of indoles, and we could not quantify individually other indoles such as indole propionic acid or indole acetic acid that play a pivotal role in modulating anti-inflammatory effects at intestinal level. We know, because the scientific literature has reported that the production of these two key metabolites that I already mentioned, indole propionic acid and indole acetic acid, is not only regulated by the tryptophan, but also by the presence of dietary fibers. And this is something that we observe in this prepreg that it's currently under review, that the presence of soluble fibers and tryptophan significantly increase the production of indole propionic acid and indole acetic acid. We also notice differences at methylhistidine, metabolite that it was exclusively found in the beef patty. And this metabolite has been associated with inflammation, but still it remains unknown the biological role of this metabolite at intestinal level. Interestingly, we found as the most relevant finding in this research the presence of N-acylamides. These compounds were exclusively found in our plant-based patties, and they have been associated with the capacity to interact with the gene protein coupled receptor that regulates gastrointestinal tract physiology. As soon as we had a map of the microbial metabolites that are being produced after specific dietary factors, then we need a model to study, to investigate the response of intestinal epithelial cells. And in the past, I used to work with 2D immortalized cancer cell lines. And well, although these models are cheap and relatively easy to use, they differ metabolically and genetically from an in vivo real-life scenario. And for this reason, we decided to use approximately 5-6 years ago, we decided to use intestinal organoids as a model to investigate gut microbial metabolite-gut epithelium interactions. For example, we use a basal-out intestinal organoids to mimic a systemic exposure, but these days we are spending so much time and effort on 2D intestinal monolayers because they recapitulate the behavior of intestinal epithelial cells in vivo. It's a system that provides you with a highly differentiated and specialized intestinal epithelial cells. Well, Carmen, my postdoc, is leading this research. She has her own research investigating lipids and gut epithelial cell responses. But as a side project, she also investigates an epicanthic metabolite that in the past we observed that regulate gut health. We expose our monolayers to known concentrations of this metabolite, and after that, by multi-omics approaches, we investigate the mechanisms in that. Summarizing the journey today, our journey today with this talk, the takeaway messages are as follows. So dietary factors such as particle size, macronutrients, phytochemicals, modulate the production of microbial metabolites that in turn promote intestinal health or a normal gut barrier. By contrast, a compromised gut microbiome or a specific dietary factors might generate a pattern of microbial metabolites that impairs the gut barrier chronically and might trigger gastrointestinal diseases in the future. Well, last but not least, I would like to thank my collaborators and my team for their extraordinary support and for the strong motivation to investigate microbial metabolites. Thank you very much for your kind attention, and now I'm happy to answer any question you might have.